We have some excellent speakers today, and I will introduce first Dr. Xiao Liu. He's from the Pennsylvania State University of Pennsylvania. Sorry, and yeah, he has done forefront research on the propagation of brain activity across species and using different methodologies, and in the past years using AI to inform fMRI. So yeah, exciting to hear your presentation.
Hi. Hello, everyone. Yeah. And yeah, this is Xiao Liu from Penn State University. Today I'm going to talk about those like the structured inflow global brain activity and its potential like the functional relevance. I have nothing to disclose. Yeah. Yeah, my research, I think it's mainly focused on the like the neural basis of those resting state fMRI. If you have ever like using this technique, you probably know that's one of the like the trickiest component is like the global mean signal, and which we call the GBO signal. Basically, it accounts for the like the majority of the like the signal variance. If whether to remove this component is gonna make a huge difference in terms of the connectivity measurements. So our group has been like working on the like the questions whether those like the global like the GBO signal, whether it's noise or it's actually the neural activity. If it is like the neural activity, what's like the functional relevance of these components? And a few years back, we using the like the simultaneous fMRI electrophysiological recording from monkeys. We show okay those GBO peaks, they're actually corresponding to very like this very clear pattern in those like a local field potential. You see those time frequency pattern, which is very similar to the something we reported like a few years back. We called like the sequential spectral transition. We saw it in the like the global mean signal of the monkey ECOG. And based on the pattern at that time, we hypothesized it's related to the like the arousal modulation. Seems the like the spatial pattern of those like the GBO peaks seems like confirm our like the hypothesis, because even though most of the brain region you see like the signal increase, but you do see very specific but very significant deactivation in those like the subcortical region, particularly in this nucleus basalis at the basal forebrain, because those are like the known to like the regulates like the arousal. But later we found it's probably more complicated. That if you look at those like the GBO peaks, like the spatial temporal dynamics around those GBO peaks, you do see a lot of times they actually behave more like the propagating waves. And also it's not random. If you look at the pattern, a lot of time it's actually between two sets of the brain region. One is those like the sensory motor regions. The other is more like the higher order, like the cognitive network, especially the DMN. So this reminds us immediately those like the PG, principal gradients, like the decomposed from the resting state connectivity matrix. And it has been like hypothesized to represent like the gradients of the like the cortical hierarchy. Yeah. If we like the project those like the resting state fMRI along this PG direction, we do see a lot of time, and those GBO peaks, it's more like a tilted band, yeah, along that direction, confirming there is like the propagating along that direction. Then we develop a method, data-driven method. We call it like the delay profile decomposition methods, and basically try to extract like the dominant propagating direction represented by this principal delay profile. If you compare this one with like the PG, it's almost identical. Just further like confirm it's actually the dominant like the direction of the propagation. And we go back to the like the monkey ECOG data. If you look at those gamma frequency bands on those SST, if you do the same thing to this prism, like the delay profile decomposition, got those like the principal delay profile, it's very consistent across animals. You got a big contrast between those lower order sensory motor region and the higher order brain areas. And you can, if you do the same thing, projecting those signal in those time position like the graph, you do see those like the tilted by those like the propagating waves. So everything in the monkey just confirmed that's not because of the artifacts from the different like hemodynamic delay, hemodynamic delays in different regions. Yeah, we feel excited about these results because those like the cross hierarchical propagating, that's really like similar to what we're seeing in the the information flow in those like the artificial neural network, because those are like a huge model with numerous like the parameters. The only effective way to optimize those like a large model, you have to do something called like the back propagation. You have to have the arrow at the last layer, then you propagate like a layer by layer back. At the same time, you update those weights. Those weights are more like the between artificial neuron. It's more like the synapse of the real neuron. So we think about whether those cross hierarchical propagation maybe just do them in the same way, like modify those neuronal synapses. But of course, that's a very difficult hypothesis to prove. But if that's the case, then we would wonder this the global waves may be related to the learning and the memory. Yeah, then we think about the hippocampal repose. That's like the high frequency field event in the hippocampus, and people already show it's highly related to the memory consolidation and also the memory retrieval. If you compare with the Jibo signal, I think they show similar the dependency on the brain states. You see much more repose during the rest and the sleep. Then about 20 years ago, I think there's a study showing those the ripple rates indeed co-modulate with the delta wave, which is an indicator of the arousal in very low frequency, 0.1 hertz. Then about 10 years ago, there's a Nature paper showing those ripples are actually associated with the whole brain global fMRI change, especially in the cortical region. This different region has significant time delay. Yeah. So if you look at the pattern, you even see some a cross hierarchical contrast. Yeah, that's why we go to this the visual coding project from Allen Institute. They use a neural pixel the recording. I think it's the large scale neuronal recording. They can record more than 1,000 neurons simultaneously from a single animal. But most importantly, they have a lot of recording in the hippocampus. So we look at the spiking data. We just apply the same methods we used to get those the propagating waves. We derive those principal delay profile. We sort the neuron according to those profile. Then we can see those very clear repeated patterns. We call it the spiking cascade. It's involve all majority of the neurons, so no matter where you recorded those neurons, but at the same time, it's coupled by those the arousal modulation. For example, the spontaneous the pupil dilation and also the delta wave modulation. But most importantly, if you identify those the repose, you can see how it's the modulator across the cycle of those the cascades. You see very significant the modulation. Because of that, we hypothesize those the cascades represents an offline process linking the arousal and the memory system. But later we found those the cascade dynamics. It's not only seen during the resting state, spontaneous session. You can see that with the continuous visual stimulation. Yeah, but basically it's much stronger than those the visual evoked response. You only see those visual evoked response after you zoom in those the visual neuron. Then you can see those the stimulation evoked response. Yeah. Then with these results, we're wondering, okay, how those cascade dynamic affect the online, the sensory and the memory process. Okay, what we do is we train the machine learning model, but we based on the neural data, we predict the stimulus ID, then use that one to quantify like the accuracy of the visual encoding. At the same time, we use like the hippocampal ripple rates to represent like the memory-related process. Then we see both of them actually modulate significantly across the cycle of the cascade, but they modulate in opposite way. Yeah, one goes up, the other goes down. But most, like recently, we just repeat everything in the human using the NSD data provided by the Minnesota group. And we do the very similar thing. We train like the more complicated like the AI model, try to decode those like the visual semantic information used to quantify the accuracy of the visual encoding. Then for the human, instead of the like the repos, we do have the behavior data. So we can look at like the user memory, like the behavior results to measure like the memory encoding, memory like the retrieval. Then we see very similar like the modulation of those encoding and the like the memory retrieval across the cycle of the like the G-boat waves. Yeah, one goes up, the other one just goes down. If you want to know more detail, you can go to the like the poster number 827. Okay, there's here's our interpretation about those results, because there's a classic like a theory about like the wake and the sleep, basically saying those two states represent very different like the functional states, like the favor the like the encoding and the consolidation like respectively. But the major like the player here is like the acetylcholine. Yeah, different level of the acetylcholine just change the connectivity between different brain region. And then, as I just show you, this G-boat signal actually highly related to the like the cholinergic system. We also show, okay, this dynamic actually modulates the sensory coding and the memory in opposite way. So our interpretation is those like the infraslow global activity represents a process like to drive the brain oscillating between like the two different function modes. One is more like the facilitated like the encoding. The other is more facilitated like the consolidation, like the memory retrieval. Yeah. We also suspect that maybe has something to do with like the very famous like the model, like the saying like the people generally like to perform best at the intermediate like the arousal level. That's just could be that at that level we have a more balanced like the encoding and like the memory retrieval. Yeah. Okay, so far I hope I convinced you. I think this like the global brain activity is related to the like the cholinergic arousal system as well as the memory function. Both of them I think are very critically implicated in the Alzheimer's disease. But the third thing that's relevant, it's a hallmark of those AD, that's the accumulation of those misfolded proton. And a decade ago, people proposed this like a glymphatic system. Basically, it's those like the CSF signal goes from the peri like the arterial space, then get into the tissue region, wash out those like the brain waste. Yeah, but if you compare this like what we know about this glymphatic system and the G-boat, they share some similarity, right? Because as I said, the G-boat highly depends on like the arousal state is much stronger during the sleep than the wake. And also the CSF is one of the like the driving force for this glymphatic system. But about like 60 years ago, I think the Laura Lewis group, they show, okay, this G-boat signal actually coupled by the very strong like the CSF movement. Yeah. So then we do some like literature research. We really feel that this global brain activity, especially those like the subcortical, like the neuromodulatory system, they have all of the capability to help with this like the glymphatic clearance. One example, for example, this like the subcortical region, we already show at those G-boat peaks, we have the very specific deactivation in the nucleus basalis, but about like the half cycle before that, we actually also see the the deactivation in the LC, locus coeruleus, those different like the neuromodulatory system, they are known to have very strong effect on the vessel, but in opposite way. For the like the cholinergic system is more like generally dilates the vessel, but the LC is more generally constrict the vessel. Altogether, they work together could modulate the vessel, creates those like the vessel motion, which could be critical for those like the perivascular clearance. And also there's like the other studies showing those like the G-bodes or those like the spontaneous like the pupil dilation actually associated with like the astrocytes activation. That could be very critical for the micro level like the CSF, ISF exchange. So because of that, we use like adding the data, we go test our hypothesis. We quantify the like the coupling strengths between the G-bodes and the CSF. We just found that it's correlated with a lot of things about the ED. For example, it's getting weaker, and as like the disease, like the condition gets more severe. But most importantly, it's correlated with like the amyloid beta, not only those recorded by PETs, but also the CSF amyloid beta. Yeah, we repeat everything in the like the Parkinson disease patients, and but for the Parkinson, we only see the significant reduction in those with like the cognitive decline. Yeah. Okay, but during that process, we learn, okay, those amyloid beta accumulation is actually not random. A lot of times they follow very specific trajectory in the AD. At the early stage, it's mostly accumulated in those higher order region. Then at a later stage, they propagate to like the sensory motor region. And there are like the different like the hypotheses, like to try to expand that. But for us, it's really similar to the propagating waves we saw, right? And then we just go test that. We found like the group of the like the subject with the early amyloid beta pathology in the cortex. It's indeed, I think, mostly in those like the high order deformed network. It's just opposite like the spatial pattern as those like the G-boat signal. But then we go to like the core subject correlation. We do found among those like the early accumulator, if you see a greater like a G-boat reduction at the DMN, then those region tends to have more amyloid beta accumulated in the following 2 years. Yeah. And this could be those like the G-boat, those like the change could be because of like those G-boat waves, because we show in subjects with a very different amyloid beta pathology, indeed, those with more like the amyloid beta pathology, their global wave actually, it's harder to get to this like the high order region. Yeah. I think that's all. I think basically, I think we show there's like the very highly structured like the global brain activity, and we show it's maybe related to the like the arousal, memory, sensory coding, and also those like the brain waste clearance. Yeah. I think that's all. Yeah. Thank you very much.